The GA provides deep insights into the blind organizing power of natural selection. Among the most doggedly persistent, widely believed, and yet patently false ideas concerning speciation and the origin of novelty are the ideas that 1. Point mutations are the ultimate source of diversity in the gene pool. 2. Recombination can only shuffle existing sequence motifs. 3. New genotypes lead directly to new phenotypes. 4. Most mutations are harmful. 5. The disadvantages of sexual reproduction almost nearly outweigh its advantages as a source of diversity. Each one of these misguided beliefs can be traced to false precepts and misunderstandings dating back to classical Darwinism, the modern synthesis, and population genetics. Each one has been boosted, at one time or another, by the endorsement of distinguished evolutionary theorists. But fortunately, due to the robustness of professional ethics, logical debate, the scientific method, and new methods of empirical discovery, they have been countered by cogent arguments. For the most part, they have been corrected for the record, but that does not mean that they do not persist to some degree in the scientific community, and to a larger degree in textbook accounts and among non-scientists. Here is a brief list of updated concepts that will hopefully replace these 5 misguided concepts. 1. Point mutations are not the ultimate source of diversity in the gene pool. In Chapter 16 of Rethinking Evolution, genome evolution will be explored in some detail. Briefly, point mutations, that is, single base changes resulting in single codon substitutions, may be the least interesting of several types of well-documented mechanisms and kinds of changes in DNA sequences during genome evolution. Also, these various types of genomic changes often synergize in consequential ways. 2. Recombination can do more than reshuffle existing sequence motifs. Recombination represents an important source of sequence novelty because it generates new combinations of shorter sequence motifs and can generate profound changes in both protein coding and regulatory DNA. This will be discussed further in the following section. 3. New genotypes often do not lead directly to new phenotypes. The concept of the developmental toolkit, a central concept in Evo-Devo, needs to be expanded to the concepts of the genomic toolkit. For example, reusable DNA sequence motifs of all kinds, including duplicated genes, as well as the concept of the genomic scrapyard, which is a suitable 21st century replacement for the earlier misguided but powerful metaphor of junk DNA. DNA sequences that are not explicitly used at any given moment in any particular genomic background or ecological context represent a potent source of materials that can be used at later times or in other contexts, and contribute in many ways to the evolvability of any lineage. Lineages that are evolvable tend to persist for many millions of years, whereas those that are not are more likely to go extinct. 4. Most mutations are not harmful. The idea that most mutations represent random changes to a complex, metaphorical, fine-tuned machine and are therefore more likely to be destructive than useful is a point well taken. However, this perspective is a limited one, because it, A, only considers the phenotypic consequences of mutations in isolation from other genomic changes, B, assumes that the mutations alter specific protein-coding codons, whereas mutations actually represent a much broader range of changes to genomic DNA that are not usually referred to as mutations, including, for example, gene duplication, Chapter 16, and C, it fails to consider the multiple weak, subtle, and combined effects of changes to regulatory sequences of DNA. 5. The advantages of sexual reproduction do outweigh its disadvantages. Sexual reproduction is the main source of variation among offspring. This variation arises during the production of sperm and egg cells, primarily by means of segregation of maternal and paternal chromosomes during meiosis, by random fertilization of one egg cell by one sperm cell, and by means of random recombination events, including exchanges between homologous chromosomes, paired contributions from the mother and the father, and other chromosomal variations. In addition to its importance as a potent source of new combinations of useful genes in each individual offspring, the presence of 2 sets of chromosomes also provides a powerful way of maintaining diversity in the gene pool while masking most harmful recessive mutations in individuals. In addition to recombining protein coding genes as well as combinations of protein coding sequences with various regulatory elements, recombination also represents a potent source of genetic novelty within genes and even at the level of individual codons. This will be discussed in the following section.